My research focuses on separations. Separations are applicable to many areas in chemical engineering, and my particular focus initially was in bioseparation. So these are separation processes applied to the manufacture of drugs in the biotech industry, and also biomedical separations. So bioseparations examples include virus clearance during the manufacture of drugs. This is a collaborative project funded by NSF, and the collaboration is with microbiology. And so we look at trying to come up with new methods for removing viruses from the purification of drugs. It turns out this is important for two reasons, not because the manufacturing methods are bad, but because one uses biological systems to make these drugs. For example, you're using cells which have been genetically modified, and most of the cells that are genetically modified today, this is done by using virus vectors, and so the cells can actually produce these virus vectors, which then exist in the product, and FDA requires that you show removal of these vectors, and that's actually a very major problem. That's one. Number two is the use of the cell culture media, which is often from animal-derived compounds. You use bovine serum albumin and so forth. And again, this can lead to possible contamination by viruses that were associated with the animal that survived. Now, there are research efforts to try and get away from animal-derived compounds and so forth, but nevertheless, it's a major problem. So this is a very interesting and exciting project that we're working on. We're trying to validate virus clearance in existing what's called unit operations. These are basically purification steps such as microfiltration, chromatography, etc. The other major area is biomedical separations, and this also comes somewhat from my industrial experience. I've done quite a bit of work looking at designing blood oxygenators, which are used in open heart surgery. And in fact, over 1 million procedures are conducted today worldwide requiring the use of blood oxygenators, so that's a lot. And these devices really are artificial means to keep the patient alive, to oxygenate the blood while the heart is stopped and the surgeon is operating on the heart. And these operations for more severe cases can take up to 6 hours.